Thank you, and thank you to the organisers for this really great workshop. I've really enjoyed it, and I've learnt a lot already, and I hope to learn more later today as well. So I'm a geneticist, and I'm interested in speech and language, and initially when we started doing this kind of stuff, people were a bit puzzled because, you know, obviously speech and language is something that kids learn, and it seems a bit of a strange thing to be looking for genes involved in it. So I'm going to now turn to this guy who's you may recognise and who's appeared a few times during the last week, not in person, of course. I'm not going to ask you who he is, but so.
Simon Fisher.
No, it's not. Yeah. Maybe I need to switch that round a bit. This is just hopeful that I will eventually grow such a luxurious beard. But he put it much better that this is Charles Darwin. He put it a lot much more eloquently than I could ever put. So he said, language is an art like brewing or baking. It's not a true instinct, for every language has to be learned. It differs widely from all ordinary arts, for man has an instinctive tendency to speak, as we see in the babble of our young children, while no child has an instinctive tendency to brew, bake or write. And I think that's just a beautiful piece of writing from The Descent of Man. And what I like about it also is that it encompasses two aspects or multiple aspects of speech and language which make them interesting, which is this intertwining of your innate endowment and your environment. You have to learn something, but it's different from other things that you learn. And of course, an instinct to acquire an art was the quote that then became the title of one of Steven Pinker's books called The Language Instinct. And he kind of missed the point, I think, a little bit, because the instinct to acquire an art is really the acquisition part is important. And we'll get back to this later. I like to just, we've heard a lot about this already, but I would just want to really emphasize for you how amazing it is what a little kid does in the first few years of their lives. So by the time they're only a few years of age, a child has already assembled this really huge vocabulary of words, and we've heard a lot about, we've heard some about how this happens, or how this might happen. And they're also mastering another really complex task, which is to be able to figure out how to recombine these words into meaningful sentences. And these are meaningful sentences that don't just relate to things that they're doing now, not just comments on what they want or what they need, but also things that relate to the past and the future, and even kind of abstract concepts. And this is another amazing thing that kids acquire the ability to do. We've also heard a little bit about how another kind of wonderful mystery, which is how a person is able to take a stream of thoughts that's sitting inside their heads and convert that stream of thoughts into a stream of sound. And we've heard on the other side the considerable complexity of being able to take this stream of sound without any kind of overt boundaries for where words begin and end, and kind of deconstruct the original thing that this person was trying to say, and come to some kind of conclusion about what the original utterance was. This is kind of a way of us taking thoughts that are sitting inside our own heads, complicated ideas that are sitting inside our own heads, and trying to kind of put them into other people's heads. And whenever I think about this, I just think this is the most amazing thing that we do. And there are a couple of reasons for showing this slide. One is just that I like to be reminded of how cute my children were when they were younger, but also I think it illustrates that this absence of a monolithic view of language, and that there's lots and lots of different things that are going into all these into these processes, and that in the first, to me, some people say this takes a long time, but to me it's amazing that a child is able to acquire this without any kind of explicit instruction. So it's like there's something about our biology that leads us to soak up language from the environment and to become proficient users in a fairly short space of time. Now, of course, this leads to the question of how might that, could something about our genes be helping this, and how might that information be encoded at a genetic level? And we're pretty sure that it's not going to be something like this, right? And we know that the individual features of language can't be coded within the genome, because we know that if a child grows up in Japan, they'll learn Japanese. If a child grows up in in the Netherlands, they might learn Dutch and some other languages as well. And if a child doesn't get exposed to language at all, then they're not going to learn it. And so again, it's a tight interplay between genes and environment. But we need to think a little bit about how genes might help speech and language to be instantiated. And this, I'm now going to show you the kind of classical model which drove discussions for many, many years on this topic. And it goes something like this. So for a long time, genes were treated as these kind of abstract entities that could mysteriously control aspects of your behavior and cognition. And lots of people had discussions about, what did was there a grammar gene? When did it evolve? All these kind of things, without any kind of embedding of this in what's known about how genes really work. So if you get one take-home message from my lecture, it should be this: there is no such thing as a gene for language, or a gene for speech, or a gene for grammar. But we need to somehow replace this with something a bit more productive. So I'm going to try and spend 10-15 minutes trying to, for those of you who know masses about molecular biology, you can go to sleep for a few minutes. Why am I showing you this? This is not my collection of multicolored socks. I'm not a strange collector of these things. So can somebody tell me what it is? Chromosomes, yeah. Anything else you can say about it? Any ideas? It's a male. Why do you say it's a male? Yes, so very good. A male, but can you tell me anything else about, it's a male, but it's a male, it's a human, right? You kind of took that for granted. But so we have these 22 pairs of non-sex chromosomes, and then we have an X and a Y if we're a man, and two X's if we're a woman. And what you can also tell about this particular person is that they don't have any kind of major, well, I can tell this, but maybe you can't, but they don't have any kind of major abnormalities in their chromosomes that you would be able to see under a microscope. And that's because we have this nice kind of color coding system that enables us to assign each one based on length, and we can see that it's all kind of intact and nothing weird has happened. And I'll come back to this a bit later on. So we get each of these chromosomes, one from a mum, one from our dad. And of course, if you dig in a bit deeper, each chromosome is basically one very, very long string of A's, G's, C's, and T's, the nucleotide code. Now, here's where it gets interesting, because as geneticists, we want to be able to decode what that stuff means. So hopefully now, just have a quick look at this, and then when it's finished. So can anyone tell me what that is? What that does? People say FOXP2. No, you can't really tell. So my point is that we can spend hours and hours of our days looking at this stuff, and it doesn't help us at all. So we need some kind of a code book for translating this stuff and for figuring out how it translates to biology. And luckily we have a very nice. This is kind of the universal code of life that's conserved across all species and organisms in the Earth. And this tells us how triplets of letters of nucleotides map to particular amino acids, and we have about 20 amino acids that we use as building blocks for proteins. And the idea here is you'll see that instead of T's we have U's here, and that's simply because, as you'll see in a minute, the code gets turned into from a T-based code to a U-based code, but it's exactly the same code effectively. So we have things like phenylalanine, leucine, serine. Each of these different amino acids has different properties. So when it gets put into a protein, when a protein gets built up, the type of amino acids that get strung together is very, very important and will determine what the protein does. So it works a bit like this. Here's a very sophisticated picture of DNA, which I spent hours drawing. And a gene, when it gets switched on, it gets transcribed into a copy that's composed of messenger RNA. This messenger RNA is basically a direct copy of one strand of the code, but with U's instead of T's, as I said. And then the protein machinery, which is, as I say, highly conserved and very, very evolutionarily old, comes along and decodes this triplet by triplet. So you get a methionine, a glutamine, glutamic acid, serine, and so on. And these get strung together in a long chain, polypeptide chain, and then something very magical happens, which is that it folds up. And it folds up into a particular shape. And the shape that a protein forms is specified by the sequence of amino acids. The function of the protein is specified by its shape. So it may become a certain shape, may make it good to be a receptor sitting in the cell membrane. There are other proteins that are channels as well in the cell membrane that can let ions in and out. There are structural proteins that make the shape of the cell. There are proteins that regulate other proteins and enzymes that catalyze reactions. So the whole of all the molecular machinery of life is specified in this way. So we have a simple linear code, very, very simple linear code, right? Very, very simple. 4-letter code gets converted into another simple code here, and that gets converted linear information into linear information in the terms of polypeptide sequences, which turns into 3D information that is actually information about function in time as well, as we'll see a bit later. And I think, again, this is one of these wonders of life that every time, if you actually think about it for a while, this is how we're made. We're made of this stuff, which is kind of incredible. So that helps us to go beyond a little bit this superficial thing that I showed you before of gene to speech and language. So we know that we have DNA in our cells, in the nucleus of almost every cell in the body. And we know then that we have, in the human genome, we code for about 20,000 different proteins of all these different types. And because of the flexibility of this system, we can make proteins that do anything that the cell needs pretty much, or a lot of what the cell needs. So every cell in your body is expressing these proteins. Now, can anyone think of a problem here? I mean, if you imagine that we've heard about different types of neurons and you're a multicellular organism with lots of cells doing different things. So has anyone, I've told you that we make 20,000 different types of proteins. Can anyone kind of think of an issue that I haven't kind of covered? Yeah.
Regulation. And then which gene is?
Yeah, so this is the problem, is because every cell's getting the whole genome, potentially it's got the code for all these different 20,000 or so proteins. People used to think it was more, by the way, but it's much lower than they expected. And these proteins, the reason that your liver cell is a liver cell and your liver cells are liver cells, your skin cells are skin cells, and so on, is because they express different arrays of proteins. We already heard about this yesterday because these kind of GABAergic and the different types of receptors that make a neuron what a neuron is, they have to have been switched on and off somehow by genes. And it's a bit like an orchestra. If you imagine if you went to a concert and everyone in the orchestra was just playing continuously, doing their own thing, and it would be really, well, some people like that kind of stuff, but I think it wouldn't be very nice to listen to. So we need a kind of we need like conductors in different cells to make sure that the cells are making the right kind of proteins, and also to change those in response to events that happen in the environment. Now, to dig a bit further into these kind of issues, we need to look to the famous neuroscientist Scarlett Johansson, who you see here, and she collaborated a few years ago with a guy called Luc Besson, and this work was related to another well-known scientific finding in neuroscience, that is that you only actually need to use 10% of your brain, and in this study, this person managed to increase her usage to 100%. Now, actually, they missed a trick here, because what's interesting about your genome is that from a technical perspective, you only about 1.5%, certainly less than 2% of your genome codes for these proteins. So you've got all this other stuff in your genome, and what's it doing? And actually, they should have tried upregulating all the rest of that, but they didn't. So maybe that will be in the next film that they make. So we do know quite a lot about what these other regions do, but there's still much to be discovered. I'm going to just spend a little bit of time on this, but I won't go into massive detail. Every gene, in front of the gene, there's a promoter, and that promoter determines whether the gene is switched on or off. So say in this case, there's a gene here that's silent that's next to this other gene. And so there are things that bind to promoters. There are regulatory proteins that their job is to switch on and off other genes. So they bind to promoters and regulate transcription. When a gene gets transcribed, it doesn't get, not all of it, actually gets into the end transcript to use to code for proteins. There's an intermediate step where little chunks are cut out. These things are called introns, and this is called splicing, because it's very much like any kind of process of splicing where you're kind of sticking together, like editing a film, for example, cutting bits and pieces and sticking them together. And it was a mystery for a long time as to why genes, pretty much every gene in the human genome is broken by introns, but many of them, vast majority. And there's been lots of speculation about why introns should be there. One of the interesting things about having introns is that you can have alternative splicing. So sometimes, for example, this exon could be spliced directly to that exon and leave this thing out. And what that means is that the same genetic code for a single protein can be spliced under different circumstances to give you different version. It's a bit like, I don't know if any of you have seen Blade Runner, but there's like now about 7 different versions of it: the director's cut, the extended director's cut, the final one. So genes can do this as well. They can make different edits of their films, if you like. So you end up with many more proteins actually than 20,000. So perhaps that's one of the reasons why we have introns, but as I say, it's still a bit of a mystery. And then there's another level where we have extra regulation, which is here, which is after splicing, the transcript, the end transcript that gets used to build proteins. Actually, they always start with an AUG, the point where you have to start building a protein. And you'll have seen when you saw this code earlier on that there are 3 triplet codes that they don't have an amino acid assigned to them. They just say stop building the protein. So actually, you're only going to make a protein that starts here and ends there. And the important thing here is that this stuff at the beginning and at the end of the transcript then actually helps to regulate how much of the protein is then being made from the transcript. And there's a whole load of new mechanisms that people have discovered involving other kind of RNAs for actually fine-tuning the expression of genes at this extra level. So that's so that's what some of the extra sequences are doing. It's regulating. It's making the genome a kind of dynamic thing that can work in your bodies all the time. Okay, so we might say then that you have these 20,000 or so proteins, and we're interested in, when we're talking about speech and language phenotypes, we're obviously interested in what they're doing in brain cells, in neurons. So now I'd like to kind of ask you guys if you can think about what kind of things the proteins might affect, what kind of features that could eventually be important for brain development and function, and how we can fill in that gap. Any ideas about those? Fabian?
Neuronal migration, for example.
Neuronal migration. Do people know about this? Maybe not. I don't know.
Receptors and channels.
Yeah, so an obvious thing would be that different proteins make different receptors and channels, so that will give neurons different properties as I said, if you're GABAergic or glutamatergic.
The oncogeny of different brain regions?
Yes. So, I mean, the point here is to build a brain, there are a number of things that have to happen. So one thing that has to happen is that neurons have to actually kind of proliferate in different places. You have progenitor cells which proliferate, and then they actually, as Fabian said, they actually move. They migrate during development into the right places that they need to be in the brain. And actually, if these things go wrong, you can get some severe disorders. And it's a really, really fundamental property. And I want to, hopefully, I'll show you. This is not what actually happens in the brain, but it's really cool. So if it works. So this slide comes from some work that Sonia was doing in dishes, where you can actually, these are neurons grown in dishes, and you're going to see how they like to move, hopefully. Yeah. So those are neurons moving towards each other, using these kind of process of neuronal migration. And this, as I say, is a really fundamental thing that neurons do during the development of the cortex. So genes are known to be important for those kind of processes. Another thing that's really, sorry.
Just out of curiosity, how many times is that
that speed it up? So what's the actual...
Oh, it's very fast. I'm not off the top of my head, I can't. But it doesn't happen anywhere near that fast. Sorry, that might be a bit misleading. But it's just to show you the principle that neurons actually can move around and do all sorts of interesting stuff. Now so let's think about other things that happen. Of course, neurons have to find the right place, but what else do they have to do? They have to wire up to the right places. So they send out early neurons are sending out these neurite outgrowths out from the cell body, and one of them has to become a nice long axon. It has to find its targets, and it has to wire up to the targets. Genes are really, really important for this stuff. This is how they, so to build neural circuits, genes are there building a kind of template of neural circuits. And now you might be saying, well, of course, I've heard about something like synaptic plasticity, right? And we've heard there are other mechanisms as well that are important for learning. But the thing that's interesting and curious about synaptic plasticity is that this involves genes, right? So you might think that your genes are kind of switched off and a bit bored after they built a brain and it's kind of done. But actually, in this kind of process, where to jump a synapse, a signal comes along that's an electrical signal, and it gets converted at the synapse into a chemical signal that then gets received at the other side. And of course, the strength of this synapse is governed by these kind of the array of receptors that are sitting in the membranes and other kind of processes. Every time a synapse, the strength of a synapse is being increased or decreased, genes are in play. And a lot of it's about local protein synthesis with transcripts that are moved into the right places. But the important thing here is that, again, this idea of if somebody talks to you, if you're talking about aspects of behavior or cognition, and somebody says to you, oh, I didn't know that was genetic, or how genetic is that? Pretty much everything is genetic because it's this very, very tight interplay between your genes being switched on and off to mediate these kind of processes of things like long-term memory, where you actually, and synapses are actually becoming more firmly established and so on. And of course, so you have so genes are helping to specify a kind of basic template of what the wiring diagram should be at some level, which then gets modulated by environmental inputs, or during the whole time is being modulated by environmental inputs, which involves genetic mechanisms. But of course, the brain is the thing that does the speech and language in the end. Now, this is already, I have to say, this is massively oversimplified, but this is the minimal kind of level that you need to have in your mind when somebody says to you that there might be a gene controlling a certain syntactic process, for example. There has to be these intermediate steps at some level. You can't ignore it. And if you're going to have arguments about if you're going to have arguments about how language evolves, you also need to bear in mind these kind of realities of developmental biology and molecular neurobiology. And even if even if we know nothing about the specific genes that are involved, we know it has to work something like this because everything else does. Yes.
Yeah, I was reading this chapter by Randy Galliston, and he was taking the analogy with the genes for the eye. So he said, we know there's a gene for an eye, so you can even take the human gene for an eye, stick it into a fly somewhere, and you can grow a fly eye. And the reason is that that gene is coding for a transcription factor that encodes for other transcriptors, and you have a whole structure.
Yeah.
So given that you have this structure, then you could imagine that there's also genes for parts of brain organization.
Oh yeah, absolutely, and we'll get back to this. And what I should say is that actually masses and masses is known about how different transcription factors work together and other factors work together to specify certain cell types and how they move and things like that in neurodevelopment. So so the point is not that there can't be genes influencing these processes. It's just that and by the way, the eye example, which maybe I'll come back to a bit later, is interesting because this is the PAX6 gene, which causes eye disorders, and it's a transcription factor. And if you inject the human version of PAX6 into a fly brain, you get a fly into a fly cells, you get a fly eye, right? But it's rare that a single transcription factor by itself can map to a whole complex organ. And PAX6 does ton of other things as well, actually. So the story is always a little bit more complicated than that, and we'll see that but there'll be lots about transcription factors to keep you happy later. So I'm not saying that it's impossible for genes to help to build certain types of circuits or help. I mean, something must be helping to map out that architecture that we were hearing about, the kind of cytoarchitectonics. But I very much doubt that anyone's going to find, for example, or I'll put my money on nobody's going to find a gene for Broca's area, for example, a single gene that says this will build Broca's area. Where am I going next? Let's think. Oh yeah, okay. So of course, going back to Darwin again and again. Here is his kind of original sketches on the ideas about how species might relate to each other. And here's a more fully fledged out picture from a few years ago. And of course, we've heard a lot about another fascinating thing about speech and language, as well as the remarkable abilities that we see in our kids and the amazing way we use language and the complexity of it. Even by itself, that would be interesting, but of course, we're interested by the fact that there's only one living creature that we know about that does this at the moment. And maybe we'll return to that later, but that places some constraints on how, if we want to find the genetic basis, how we're going to actually do it, because people who are interested in eye development, they do lots of work in other species on eye development, and they've got lots of systems and ways of finding the genes without necessarily needing humans. So my question to you, because I need to, like Anne, I need to write grants and I'm kind of trying to get some ideas. How can we find genes that are important for speech and language then? And people who already do this stuff, stay quiet, because I know there's a couple of you here. So just any crazy ideas about what we might do? Yeah, and I'll talk quite a lot about that. So one approach is to look at, instead of looking at we've heard quite a lot about adult pathologies, we could look at developmental pathologies and try and find the genes involved. So that's one idea. Any other ideas? Yeah, and that's coming later. But so that I think would fit in with this idea of using developmental disorders. And I'm going to spend quite a lot of time talking about developmental disorders and where we can go with that. But I was curious as to whether anyone had, because we've been doing a lot of that, and it's going quite well. But I'd like some new ideas. So, any other thoughts about? I mean, think about that's one way we could go, is to look at developmental disorders. Are there other things that we could do? Think about something. Oh, go on, Anne.
Well, perhaps using individual variability. When we saw that genius who spoke 60 languages, you said, "Oh, I'd like to have a look at his DNA."
Yeah, nobody was paying attention. So, yeah. So this is something that nobody's yet done, but I think would be really interesting, which would be to go to the other extreme and try and find are there things that make people really, really amazing at aspects of language. It could be that they learn 60 languages with ease, or it could be that. I mean, a more kind of random example might be if some people are extremely eloquent with the way they can use language and seem to have really. And then, there are simultaneous translators. Maybe this is something a bit different from what goes on in normal language, but I think it could be interesting to know why a lot of people go to these schools for simultaneous translation, and not everyone can make it through the course and come out of it at the end. So you could have a kind of be able to look at the people who'd failed and the people who'd become these wonderful simultaneous translators. And any other ideas that might relate to what I said here about us being a kind of outlier? Yeah.
We could go out and look for any organism who speaks and then compare with people. So maybe there's a worm out there who talks, but we didn't find him yet. And then we could compare genes.
That's an interesting idea. Well, honeybee language maybe. I think one, and I'll return to this at the end, but one idea that people had early on was that if we knew what the genome of a human was, and if we knew what the genome of a chimpanzee was, we'd be able to put those together, compare them, and figure out what the genes are that are important for making us human. and amongst which there should be something to do with language, right? Because that's one of the key things. Now, I'm going to come back to that right at the end of the talk if we have time. But I just wanted to give you a flavour that there are limitations, right? When we it's a tricky question to be able to figure this stuff out, but it is doable. And the thing I'm going to the thing I'm going to focus on is this idea that we use the human population as a kind of natural experiment, and this encompasses all the ideas about pathology and individual variation. The idea is simply that within us there is variability in our abilities to acquire speech and language. And there's been some resistance. I think people haven't thought about this so much in the past because there's been such a strong focus on universals of language and the fact that everybody can do it, and the idea that anything, any variation must be kind of trivial beyond pathology, which say Chomsky linguistics would accept the fact that pathology is okay, but everything else is basically we have exactly the same abilities. Everyone has the same competence. I think that's unlikely to be true. So the idea there, so in human genetics we have these very, very powerful tools which can look at the phenotypic variation. So phenotypes are the sort of the things that we observe in people, the things that we can measure: height, weight, how good we are with language, or different aspects of language, how good our memory is, things like that. So these are our phenotypes. They vary in people. We also need to then know something about how genes vary. Now, as I say, we don't actually know that much about how speech and language skills vary between different people, and beyond things like how many languages they've learned due to their environment, things like that. But we're learning more and more. But we do know a heck of a lot about how genes and genomes vary. I mean, and this is especially in the past few years with the advances in genome sequencing tools. We really understand a lot about the differences between everyone in this room, for example. We don't necessarily know what it all means, but we have catalogues of variation. And these variations occur at striking frequencies in the population and at in different kinds of ways. So one way is, here is, as I said before, here is a normal male. Now, in some cases, there are major changes to chromosomes. These are quite rare, but a bit more frequent than we realized. And they may actually change the structures of the chromosomes that affect the way that genes work. So one example here is something called a translocation. So this might be, say, chromosome 1 and chromosome 5. And what's happened here is a chunk of chromosome 1 and a chunk of chromosome 5 have been kind of exchanged in an aberrant process in the parent of some kid. And then the kid basically then, all their chromosomes look like this. All their chromosome 1 and chromosome 5s that have been translocated. This is called a translocation. And the point here is that this person might have all the genes that everyone else has laid along the chromosome. So it really depends where this breakpoint has occurred. Because if it's in the middle of a gene, of a protein coding gene that's important, it might actually inactivate that gene and stop it from working. Or it could even place kind of one gene under the control of another gene, for example. So these kind of translocations can sometimes be absolutely benign and people can carry them and not even know that they've got them. But sometimes they can actually mess with the gene and cause a disorder. Similarly, sometimes people have these big deletions that actually take out a whole chunk of different genes. There are examples of things, and also you can sometimes have other things like duplications. So those of you who've heard of Williams syndrome, this is a disorder that relates to a part of chromosome 7 losing a whole chunk of different genes, a large number of different genes actually. And of course you all know about Down syndrome, trisomies, where you actually, trisomy 21, where you have an extra copy of the whole of chromosome 21. So sometimes copy numbers is important. And there are other types of big chromosomal rearrangements, like inversions and so on, and we can see these things under the microscope, and often they lead to pathologies. Right at the other extreme, we can have really, really tiny changes that just to a single nucleotide. And the consequence of this single nucleotide change depends on where it is, and on where it is and what type of change it is. And the thing to go back to here is this triplet code that's conserved, that's basically used by all the protein building machinery on Earth. And here you can see that there's a bit of redundancy in the system because you can potentially code for 64 different types of amino acids, but you only need 20 plus these 3 stop signals here, this one here, UGA, and these 2 next to it. So in some cases, certain changes to the genome will not affect the amino acid sequence at all because of this redundancy. But in other times, it can mess with the sequence. So if we go back to this interesting gene here, that building a protein, you can imagine that if you had a mutation here from GAG to GAA, that changes glutamic acid to a glutamic acid. So it's exactly the same. So you might carry this variant, and many of us are carrying all these variants that don't change our protein sequences. And they're called synonymous variants. But you can imagine if we take the same point and instead we change this earlier G to an A, somewhere else in the code, it goes from being a glutamic acid to a lysine, which is an amino acid with different properties. This can change then the shape of the protein forms, and depending on how well it can still retain its function, it might actually lead to a severely dysfunctional protein or something that doesn't work quite as well as it would normally do. And again, we're all carrying these kind of things, these kind of non-synonymous mutations. There's a number of these that are really, really well characterized in certain brain-related genes that, like 50% of us in the room would have them. And another thing that can happen, of course, is that if you change this same position, exactly the same position, we change it to something else, it sticks a stop signal, right, where it shouldn't be. And you can imagine this protein actually goes on a lot further. So this person is now making, instead of making this big protein, they're making a tiny little truncated thing. And often there are processes that mean that you don't even make that at all. And this is called, in this case, it's called a stop gain because it's gaining a stop, or sometimes people call them nonsense mutations. So we're interested, so you can catalogue in each person the array of missense mutations and nonsense mutations that affect their protein coding sequences. And people are doing this with a whole genome, whole exome sequencing now. Or you can look at one individual gene that you're interested in and see this. Going back to our well-known neuroscientist, Scarlett Johansson, remember she'd increased her brain usage to 100%. In this other 98% of the genome, this is where all this regulation is happening. And again, we know a lot about how variants, less than protein variants, but we know quite a lot about how regulatory variants might be important. So remember I said that at the front of every gene you have a promoter, which switches that gene on and off. Now you can imagine that if you had variation in that promoter, it might actually change how efficiently that gene is being switched on and off in certain circumstances. And that could lead you to have, in some cases, have a disorder, but most of these effects are quite subtle. Remember that we have this splicing thing where introns are cut out, and there are things called splice sites that tell the machinery of the cell where to cut these things out. And you can also have mutations and variants that interfere with that process or affect the efficiency of that process. And finally, remember I said that in these untranslated regions, UTRs, these are important for regulating how much protein is made in extra mechanisms. So again, you can have variations there as well. And we're beginning to be able to read off those variants and try and say something about how they affect protein function. So to sum up, we know lots and lots now about this, and we have catalogues of variation from normal people and in all sorts of different disorders. And this ranges from these kind of big scale chromosome rearrangements, translocations, deletions, duplications, inversions that were mainly interesting for people who were working in pathology and in clinical genetics for many years, but might also be interesting in other things as well. There's something that I won't spend any time talking about, but just be aware that there's something called copy number variants, which is smaller deletions and duplications that you don't see under a microscope that might actually be quite subtle, but they can lead to duplication of genes or loss of individual genes. And these are just kind of smaller versions of these, if you like. And then right at the other end, you have single nucleotide variants. You have some other stuff going on in between this level. And the single nucleotide variants might be missense or nonsense mutations that change the actual proteins, shapes of proteins, or knock them out completely. and regulatory variance that affects those other processes. So what we want to do is to be able to relate these kind of variations in people to the variation in their phenotypes. If we find a correlation between a particular type of variation, naturally occurring variation, or maybe in a pathology case, and a particular phenotype, then that suggests that there might be a causal link and we can follow it up. And here there are certain types of variations that, as I said, everyone in the room, or 50% or 50% of the people in the room, up to 80% of the people. So there's common variation, and there's lots and lots of it. But there's also very rare mutations, and they might be. You probably carry your own unique private mutation that nobody else has, right? Those are very, so you're special. So, well done. The issue as well is that there's a range of effects, and it tends to be that the most common things, common variants, often don't have as dramatic effects, which you might not surprise you, because when a rare variant comes along and it has if a variant comes along and it has a really severe effect, and you don't develop properly and you're unable to have kids, then it's gone from the pool, if you like. So rare variants, or the severer variants, tend to be rarer. Okay, so that's the kind of shape of genomic diversity. Did you want to?
Yeah, I want to come back now to the mind the gap slide.
Yeah.
So you have varieties of spice of life at the phenotypic level,
Yeah.
and now at the genetic level.
Yeah.
And we have to
Yeah.
mind the gaps at all
Yeah,
different
absolutely,
steps. Now,
yeah.
the research program is starting to get really complicated, right? And I have a many-to-many problem, because you don't know what the relevant variations at this level, and you sure as hell don't know what the relevant variations at the phenotype level, because we don't know what we're looking for.
Yeah. So
It's pretty tricky.
No, I don't. No. So what you're doing is you identify phenotypic variation that you think is interesting, and one of the ways of defining how it's interesting is I'm getting ahead of myself, but there are ways of finding out whether this phenotype is likely to be more likely to have genetic contributions than other phenotypes. You need to know that your phenotype makes sense from, and then what you're trying to do is unpack the genetic variation that is correlated with that phenotypic variation. And the point here is that is not the end game. That is the starting point for filling in the gaps, and that's what I'm going to try and show in the next 15 minutes.
I guess the kind of hands-on research question I have is to say, look, let's say we have a particular phenotype that we think we can get some purchase on, like non-word repetitions.
Yeah.
The step one before that in the mind the gap is, let's say, neural circuitry, broadly speaking.
Yeah.
Now the question is, that mapping also is not one-to-many or many-to-one. There are many ways to actually get success or failure on that. So where do we look for that?
So if I get time, I'll get to the issue of actually, because there are ways also of mapping genetics directly to variation in brain phenotypes as well. And in fact, what's happening is people are doing that as well. So you can try and sort of go inwards, and you could potentially also look for variation but you might have to use other organisms for relation. And actually, I think this is done. So look for variation at the level of neural circuitry, but you can't do this in humans, and genetics. So you can you can be informed by those things, but in the initial idea is that you get a kind of window into the biology by finding these correlations that may not. Yeah, you have to be prepared; they may not pan out. But this is how modern human genomics can try to get insight into the biology of these traits using existing variation and using pathology and things like that. And that's what I'm trying to get at. So yes, keep in mind still this mind the gap slide. But I think hopefully what I'm going to show you today shows you that you can make steps in that direction.
Can I ask a slightly?
Yes.
So you can say variety is the spice of life, but even at the level of the phenotypes, it's not endless variety, right?
No.
So clearly,
Constrained.
normal repetition, you can be a bit better, a bit worse, but everyone, lo and behold, Homo sapiens
Yeah.
can do non-repetition. Likewise here, I guess there is a lot like there's variety in the genome, but that's, I don't know, is there any percentage to be set? What's the variation space here? Is there anything that we know about that?
Well, every point mutation that can happen will happen in the genome.
Yeah, right.
So it's a bit difficult because it will happen, but you know, if it's bad, it will. If it's incompatible with the, so there must be some. There are constraints, but they happen at the level of they kind of get weeded out. So it's a continually, because the genome's continually in flux, right?
So that means that, let's say, certain I assume key genes. That,
yeah, sure.
That
Yeah.
if they're not there, then we'll.
Yeah, then you're
the gene.
then yeah.
These might be crucial also for
Yeah.
those you don't pick up with your variety
No,
of variety.
no.
Kind of stuff.
So the point here is that we can only see. So say there was. This is a really important point. So say there was there was in fact something that is actually fixed in the population, such that you never get any variation, because if a gene mutates in this, then as soon as your gene is gone, you can't survive. Then you won't find them with these kind of techniques, unless you look at aborted fetuses or something like that. Yeah, but I would argue, and what we're seeing now is that pretty much every protein coding gene it does vary. So I think almost every gene because we have catalogues of the different variations that have been found. So in some cases that leads to a very severe disorder, but I don't want to get too bogged down on this because I want to get into the actual science of the
cross-species comparison.
Yes. So that's but I'll get back to that. I think yes.
Is there a way experimentally to hope to determine if there was an important mutation 100,000 years ago to identify it? And related, what do we know in this regard about the Neanderthal genome?
Yeah. Can you hold that thought, because that will be that's kind of the last part after the break. Actually, quite near the end, I want to get back to that issue, but exactly that issue and there are, and the answer is yes. But I'll show you how at the end. Okay, so now I want to spend 15 minutes because I started about 22, so I want to take us to the end of the hour by kind of turning you into gene hunters, and I'm going to focus on the easiest case. And partly also because it's been the most successful for us, which is to look for families showing simple cases of pathology where they seem to be inherited in the family. And the way you can use the family structure to be able to pin down the gene that's gone wrong if you assume there's a gene gone wrong. So I'm going to show you how this works. So here is a nice little family here, to get used to how you should interpret these. This is a girl, this is a boy, and they meet, they fall in love, or maybe not, I don't know. And then they have a couple of kids. Yeah, whatever, but yes, they've had their kids. Now let's imagine, I'm going to simplify this by just having one chromosome here. And remember, each chromosome has the same sets of genes along it, right? But you might get a slightly different version from your mum and from your dad. So each of these people has two different versions of chromosome. Let's say this is chromosome 1. And to help you identify them, I've coded them with different colours. Now, they have their kids, and the key thing here is that they can pass either of their chromosomes on to their kids. So this girl, she's got the green copy from her dad, and she's got the red copy from her mum. This person's also got the red copy, but got the black copy. Very exciting. I can see how riveted you are. Now, this is what happens next. They go out off into the world and they find their partners, and as it happens, both of these kids decide to have their own families, and particularly these guys are doing well. They've got 5 kids, and these guys have got 3. And again, another copy of chromosome 1 is coming in here, another 2 copies here, and when this family, when you get to the third generation, there's even more shuffling around. There's a blue copy here, here, here. So you can see what's happening is by the time you get to the third generation, you've got all these different... now again, it's the same sets of genes, but because people can have different variations in each of these genes, the particular variations that these guys carry are a mishmash of ones that have come from previous generations. Now imagine, if you will, that this is a family with an interesting speech and language disorder, which we might talk about a bit later. So these people are affected with a speech and language disorder, but the others aren't. And what a geneticist, if they look at a family like this, they might go, this is really curious because about half of these kids are affected, half are unaffected. It's seen in every generation. It could well be that there's a single gene that's damaged in some way that's causing this disorder, and it's and we want to know what that gene is. Now, what I'd like to know now is, if you're just looking at this, is there anything that weird? If you see something interesting about this, put your hand up. Right, okay. Some people not yet got it. I'll help you out a bit more. So if you look at start looking at the chromosomes that came in at the start, because the idea is that on one of these chromosomes, presumably on a chromosome here, if we're lucky, there was the damage, and that's been transmitted to, by chance, happens to have been transmitted to all the kids. So if you look at the black copy of the chromosome, it doesn't co-segregate. If you look at the green copy, it doesn't co-segregate. But if you look at this red copy here, you can see it's found in all the affected members, none of the unaffected members. So this would be a nice clue that somewhere on that chromosome is the gene that's gone wrong. This is called linkage analysis. And it's been a very powerful tool for finding all sorts of different important disease genes, cystic fibrosis, muscular dystrophy, all the Mendelian genes, or a lot of the Mendelian genes were first looked at using this kind of approach. Now this might not be so helpful because all this can tell us is that it's on chromosome 1. And chromosome 1's a big chromosome, right? And by the way, I should say that the way that geneticists are, we can use genetic variations that aren't functionally important as kind of signposts for different chunks of the genome. So we can kind of almost color code in the lab how these chromosomes are being transmitted. Oh yeah, so here's just showing you that there's where that little X is where there's a disease gene that's causing this disorder. Now, luckily, biology's helped the gene hunters by having this thing called recombination. And recombination is where, when you have a maternal and paternal copies of chromosomes, they actually have a bit of exchange between them. So each generation, chromosomes get shuffled around. And crossing over events tend to happen randomly through the genome. There are certain places that they're more likely to happen than others. But this shuffling around, this recombination, means that you are effectively a kind of patchwork. Each of your chromosomes is a patchwork of the allelic variation, the different versions of genes from your grandparents and from your great-grandparents. Which means that the color coding here gets a bit more complicated. So you can see here, here's mum here. There's been a recombination here, which means that this person is inheriting a red chromosome kind of stuck to a blue chromosome, a light blue chromosome. Now this helps us because this means that we can figure out where in the chromosome the damaged gene is. So again, if we go back to this family with this interesting disorder, if you look really carefully at it, you can see, now we look at the red chromosome again, and you can look for the areas of the red chromosome that are shared by all the affected people but are absent from the unaffected people. So what that tells us, in fact, is that because this person has the red chromosome up to here, but they haven't got the disorder, and this person has the red chromosome all the way down to here, but they have got the disorder, that the disease gene must lie in this interval here. Okay. And then finding the gene after that, I'll get back to this later on. Now, could this actually be done in real life for speech and language disorders? Well, it can. So here is the KE family. This family were identified by a group working in London, led by Farron and Varga Kadem at UCL, at the Institute of Child Health. And what we have here is 3 generations. So it's a bit like the family that I showed you before, but this is expanded out. 3 generations These are affected individuals. About half of the people are affected and half are unaffected. These kids are growing up together, and yet, look, some are affected with a severe, well, we'll hear what the disorder is in a minute. Some are escaping unaffected. And all you need to know here is really that these lines mean that the person was deceased at the time that we drew this diagram. The stars indicate people that we couldn't get DNA samples from. But other than that, this is just a standard way of showing a multi-generational pedigree. So, as I say, this family were identified because there are several individuals who were all attending the same kind of special language unit in London. And then when the clinicians drew back, sort of reconstructed that, they found that the problems were occurring in many, many more kids. And then when they drew the family tree, the geneticists were going, oh, this looks like, you know, one of our a classic single gene disorder, which is really still one of the most unusual cases where we've got such a clear-cut family tree. Now, of course, you don't know whether there really is one gene damaged or not at this point. But what happened next was a correspondence to Nature by Myrna Gopnik, who's a psychologist. And she said that the kids in this family suffer from something called feature blindness. Put your hand up if you know what the WUG test is. WUG test. Okay, so you're not allowed to try this, but here is the WUG test. That's a WUG, okay? Now there's another one. There are two of them. There are two WUGs, very good. Okay, so you don't have a language impairment, and that's in English, right? So what Gopnik said was, when they do this test, the kids in the family, they fail. They can't do it. She also said, if I say, you can do a thing where you say, every day David blicks. Yesterday, David blicks, right. So she said they're also crap with that task. She said they know books and book, right, because they've learned it as a lexical item. They're fine with stuff that they've learned as lexical items, but give them new material, they can't generate these rules. They have a selective deficit in the accurate usage of syntactical semantic features of language, such as the significance of number, gender, animacy, proper names, tense, and aspect. They lack a general rule for producing plurals. Okay, that to me sounds, knowing what I know about how genes map to biology, I think that's quite a strong claim. Now I want you to, if this works, we'll see. I want you to listen to some of the kids from the first generation of the family talking.
They are short. It's this one. They are 6. Their name is Tommy. Their is Howard. That's Howard was 6. That is reading. I live in evening. Like the cow, I live in evening. And I am like.
Did everyone get that? These are the kids, and these recordings were taken around the same time that they were doing these tests with Myrna Gopnik. So, and what they were saying was their name, where they live, how old they are. So I'll play it again, hopefully. So, I'll play it again, and so this is Stuart.
They are short. It's this one. They are
He's 6. This is Zoe.
Their name is Tommy. Their is Howard. That's Howard was 6. That is reading. I live in evening.
And Carol's probably the most
cow,
easy to understand.
I live in evening. And I am like.
Okay, now, does anyone think these kids have feature blindness? I mean, they might do, right? But they also have quite a severe speech problem. And this is, we've heard already about speech apraxia. So this is kind of like, there is a developmental version of speech apraxia. It's called childhood apraxia of speech. It's also called developmental verbal dyspraxia. And it involves difficulties in learning and producing sequences of mouth movements. It impairs speech development. This is data from a paper by Kate Watkins here, and Nina, who's sitting over there, and Farina, where they were looking at a whole range of different things in the K family, including this, which seems to be the most striking problem that they have, which is really comes out when you give them word repetition tasks and non-word repetition tasks. Well, non-word repetition is always the best for it seems to always pull out interesting things. But and what you can see here is simple nonsense words, complex nonsense words, the number of syllables that they're having to repeat. And this is based, I think, on about 13 or so affected members, 30, which are these guys here, 13 unaffected members, these guys here, and about 10 people with various forms of aphasia from Nina, which is these guys here. And I think quite a few of them had adult speech apraxia, I think, if I remember correctly. And the thing to see here is that the kids do worse as or the family members do worse as the utterances get more complex and as they get longer, which is a classical feature of speech apraxia. And another thing I want to do here is, this is from Kate. This is Kate doing some work with one of the adult members of the family, and just asking them to repeat some real words 5 times. So she's just asked this person to repeat a word 5 times.
Impossibility.
Impossibility, 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 impossibility.
It's not bad, right?
And catastrophe.
Catastrophe, catastrophe.
Catastrophe.
It's a tongue twister, she says.
Catastrophe.
Catastrophe. Catastrophe.
Yeah, just try and say it 5 times.
So that Kate's a lovely, I have to say she's a lovely person. That makes her sound like, just say it 5 times, go on! So that's even a word that you know, it's in this woman's lexicon, and yet she's having a lot of trouble. She did say impossibility fine, but with one error. But they really fall apart on nonsense words. So that gives you an insight. You know, they have a lot of speech therapy and they get better. Now, what else would you like to know about this family? If you want to know, we want to know a little bit about. Shouldn't have seen that. But what else do we want to know? Go on. So you mean how long lasting is it, or what?
Yeah,
I can tell you that. I can tell you that this persists through life. But you can hear that you know, some people go like, oh, this family that can't speak. They do all right, but they would always, if you challenge the system in this way, like giving them a nonsense word to repeat, they always fall apart, even after years. So it can be kind of trained up to a certain extent. But what other things would you like to know? Because, go on.
I'd love to know if there are non-speech attributes of their behavior that map with their speech.
Right, exactly. So non-speech. So what we want to know is this modality specific, right? Because what you might say, okay, let's go back to this test, right? If I said to that one of those kids, what is that? And they kind of go wug. Maybe they just missed saying the S or something like that, you know. So there's a compound here. So you want to know whether this kind of any deficits that you see, you want to know whether they have problems with spoken language, written language, how is their expression of language, and how is their reception of language as well. And I just show you one thing, which is a different type of language task, but this is used a lot with kids with specific language impairment, and this is testing reception of grammar. So this is called the Trog test, and what you get here is sets of pictures. You do not have to say anything in this task. You don't have to to produce any speech output. It's purely point to the right picture. Okay, so do people know this task? Put your hand up if you've... Okay. Okay, here we go. I'm going to show you a word and you need to tell me which. Pencil on the shoe is grey. Yeah. Yeah, so you would just point there. Very good. Well done. Brilliant. They get harder. And if you couldn't do that, then come and see me afterwards. If you speak good English when you couldn't do that, come and see me, and I'll take some DNA from you. But the point here is that the kids in the family do quite badly. I mean, not as badly as they do on these speech tasks. They do badly on this kind of test. But what I really want to emphasize is they do badly on spoken language. They do badly on written language. So written tests of, you can do things like fluency, verbal fluency.
Has it been tried to teach them sign language?
Yes, not well. One of the kids has been learning sign language, and he's being assessed for actually how well he's doing with sign language. It's not, it would need to be probed to the same level as.
So it's modality specific?
No, we don't know because we don't know if it's modality specific because the sign language hasn't been investigated in the same way that the spoken language has. So as I played to you already, they can speak, and especially in adulthood, they can they can give you grammatically normal sentences, and the point is, if you probe the system, you find that they underperform compared to the unaffected on all sorts of different aspects of language, expressive and receptive. They also, by the way, do have access to, they make over-regularization errors, which shows that they do have access to if there is to a rule for plurals. Sorry, go on.
Yeah, I was wondering, Nina was telling us that when you lose speech, you also lose the ability to solve complex problems. So how is there...
I think that's possible as well. Well, so that's exact. So here's another thought, isn't it, that I was kind of heading towards, which is you want to know their general cognitive behaviour, right? So, and what's interesting here, this isn't a very bright family, and many of the people in the family have IQ scores that are sort of between 80 to 100, kind of low level. So this is occurring. This disorder is occurring on a background of generally low intelligence. But there are unaffected individuals in the family who have low nonverbal IQ, and there are affected individuals, some of them, who have normal nonverbal IQ despite having these speech and language problems. Now, what then you can see if you read this paper by Kate, which I would recommend to because it's the most detail that you can get on the phenotyping of the KE family, is that when you break down, there are these different verbal cognition tasks and these different nonverbal cognition tasks, and they do badly. They do particularly poorly on tests of verbal cognition. On all the subtests of a verbal cognition test, they do badly. On tests of nonverbal cognition, they don't do terrifically, but there's one particular test which has gone out of my head at the moment, but it's one that arguably relies most heavily on kind of processes that are verbal related. So I wouldn't actually call it a nonverbal test at all. I'll look it up in the break and I'll tell you about it afterwards. But the point is that this seems to more profoundly affect verbal than nonverbal. But it's a complicated, messy disorder, as almost every neurodevelopmental disorder is.
It's the opposite from the Williams syndrome kids. They also have some language problems, but that's less severe than the other.
Yeah, Williams syndrome is also an interesting case. I could give a whole talk on that because Williams syndrome has fallen into there is a sort of pastiche of Williams syndrome that it's much more messy than the classical story in the literature that Williams syndrome have preserved language, but other kinds of aspects. Yes?
How do they deal with being, in the focus of so much research? I mean, it must impact on their life tremendously. And
Yeah. I think
can we go back to this at some point?
Yeah. I think there's a whole big discussion that one could have. I think Farron has been very good in that she's kind of protected them to a large extent from media and from and actually so so that means that there's been less work, I think, done on them than we would like to, really, because we'd like to now revisit a lot of these questions, and it's hard because they've been studied so extensively that to do more work is difficult. Yeah. Yeah. Yeah, that, I mean, it's looking for kind of dissociations is interesting. Is that what you're saying? Looking for places where relative strengths.
I mean, whatever the preserved functions are,
yeah.
They might be achieved in a different way.
Yeah. Yeah, it's kind of compensatory mechanisms. Yeah, that's possible. But all I want to so I think we should take a break, but all I want you to be thinking about during the break, and we probably don't have much time to discuss it, but I mean, would you consider they have a specific language impairment? Would you even say they have language impairment? And this, I mean, I think they do. I think they have a speech and language disorder, and I think the disorder is what it is for me. But sometimes people get very hot under the collar and want to argue with me about whether, this is all secondary to a motor problem or whatever. And I'll tell you in the next half where my direction goes, how I approach it and how I try to. But maybe during the break you can think a bit about what we can briefly talk about it when we come back. So should we come back in 20 minutes? Is that good? Okay, thank you.